Welcome everybody to our webinar of today. Let's wait a bit still for people to join. We have 14 attendees at the moment. Some are logged in.
So now you can hear me.
Yes, yes, I can
but
hear
cannot see me.
No,
Why
I
okay, I'm new with this platform.
Yes.
So then for the discussion part, do we have interactive discussion maybe?
Yes. I will need to unmute people. So I have unmuted them. So they are now on self-unmute. They can also raise their hand to ask questions. But they cannot share their cameras.
Okay.
Good. Okay, welcome everybody. Let's wait another minute and then we can start. Okay, welcome everybody to our Work4You webinar on AdversOmics. It's a new topic that we want to start with Work4You. I'm very happy to present to you as a speaker Fariba Ahmadizar. She's also part of our group at the University Medical Center Utrecht in the group of Miriam Stokenboom. She's an assistant professor, pharmacoepidemiologist with a huge background on electronic health records research. And I'm very happy that she wants to lead this group and this team and introduce AdversOmics to us to see if there are risk factors for adverse events following vaccinations that could be associated with genomes and genomics. This is very new and very exciting. The webinar now will work like this, that if you have a question, please raise your hand. I will also unmute you, so you can also try to speak up. Sometimes this is working or I need to unmute you again. It is good to raise your hand if this is not working. We will have first a presentation and then we will have time to discuss and ask questions. So if not urgent, please let's finish the presentation first and then have the questions. If there are urgent questions that you feel that cannot wait until the end of the presentation, feel free to interrupt. Yes, is there anything, Fariba, you want to?
No, thank you very much, Daniel.
Yes, very good. Thank you.
Thank you very much for this nice introduction, Daniel. Hello, everyone. My name is Fariba Ahmadizar. As Daniel introduced me, I'm an assistant professor in Miriam's group working on vaccine safety and effectiveness study, and very happy to announce our new initiative in terms of AdversOmics study to unravel biological pathway contributing to adverse events related to COVID-19 vaccines. For this presentation, I would like to start with a bit background and previous studies in terms of genetics related to medication and vaccine, move on to AdversOmics paradigm and objectives and study design of our study. And the last slide of my presentation will be about the plans ahead of this study and challenges that I would like to discuss with you guys about especially about the challenges to get started with this interesting project. Just to get started, as a heads up, I'd like to start with the latest report from WHO about COVID-19 infection and mortality rate. As you can see, based on this picture, despite that the hectic situation is coming down, still the rate of mortality is high. There are lots of studies in terms of biomarker discovery in relation to disease etiology, prognosis, and intervention so far, which are great. When you look at the PubMed or just simply Google COVID-19 infection or look at the grant application, you can see that people are more and more being interested in this area to help and to inform science to prevent the disease itself, also to prevent COVID-19 infection complications. After a few months of pandemic, different vaccines have been formulated due to urgency. I believe that companies did not follow the standard procedure. Maybe they did, but for the testing efficacy and safety of vaccines, people got vaccinated, and I personally think that we're all among a big running trial where we will get the results probably somewhere, maybe not soon, but we will be informed. Anyway, to date, we have 4 COVID-19 vaccines that have been authorized in most countries, including microRNA vaccines. As you can see here, Pfizer and Moderna, and viral vector-based vaccines, including AstraZeneca and Janssen. These vaccines are designed to train the immune system to recognize COVID-19 spike protein, which binds to host receptors and will lead to increasing antibodies, IgG and IgA, against the COVID-19 spike protein. Trials already ensure that the benefits of vaccinations outweigh the risk, but despite that, they show that the vaccination is safe, but still, like other medication, we expect some adverse events. Among these adverse events, we have serious adverse event of special interest that we're interested in this project, including Guillain-Barré, thrombocytopenia, and myocarditis, pericarditis. There are lots of studies so far in terms of ongoing studies in terms of safety of vaccines, COVID-19 vaccines. We're all involved in different projects where we study adverse events related to vaccinations. In a recent study within the WAC20, we showed that the increased rate of myocarditis, pericarditis, where the rates were significantly elevated in individuals in age 12 to 20 years. So these results showed the effect modification of age. It's not only age, there are many different factors that are associated or modify the association between vaccination and adverse events. So among these factors that are very important, we have genetic factors, which is the special interest and which is the topic of this project. Just to give you an example of previous pharmacogenetic studies, there are a lot of pharmacogenetic studies have been done so far. We have just giving you example of my previous studies. I just picked up one which is about NOS1AP gene variants associated with paradoxical increase of QT interval shortening effect of digoxin, which in this study we showed the effect modification by genes in relation to the effect of digoxin in special population. There are also a lot of studies that finally show that gene-drug interaction and lead to sometimes to guideline-based amendment in genetic testing which is required for some medications like warfarin or abacavir. When it comes to adverse drug reactions, we have a terminology which we name it as AdversOmics, which is the extension of pharmacogenomics as a new paradigm for vaccine safety and design. This is the application of immunogenomics and system biology, which we understand the genetic drivers of vaccine adverse reactions at the molecular level. In these studies, we will look at genetic regulators of the immune system and inflammation to predict susceptibility and to prevent the likelihood of adverse events. There are some genes already proved by previous studies that are related to adverse events related to medications, especially vaccines. Among them, we have human leukocyte antigens and also interleukins. I selected one study about the vaccine associated with reactogenicity and genetic testing for this study. It's not so much studied when you look at Google and when you look at PubMed to try to find previous studies about AdversOmics, but just a few studies, and I picked this one as an example. It's about genetic basis for adverse events following smallpox vaccination. In this study, 2 independent clinical trials among healthy vaccinated people have been selected for this study. Genotyping was done with the same panel of 1,400 SNPs. The first trial included in this study has been used as a discovery panel. Among these 1,400 SNPs, which were candidate genes associated with reactogenicity, 36 SNPs and 26 genes were associated with adverse events. The second trials has been used as a replication panel in this study. Among these 36 SNPs already associated in discovery trial, only 3 SNPs were associated with adverse events. And this study showed the SNPs were in interferon regulatory factor genes and showed the role of immune system in the adverse events related to this specific vaccine. So now let's move on to the objective and study design by just giving a brief introduction and background of previous studies. We want to build up AdversOmics pipeline within Vac4EU and Rock20 platforms in collaboration with GVDN. The plan that we have in this initiative is we want to form a global network of researchers to study the role of genetic variants in COVID-19 vaccine-related events. The objectives that we have in this study is to conduct a large genome-wide association study. In previous example, I showed you it was candidate gene approach. They just selected some SNPs that already showed association with adverse event, but ours have more power because we want to look at the whole genome. The genome-wide association study is the approach in this study to define common and rare genetic variants associated with adverse events of special interest, which is Guillain-Barré, thrombocytopenia, myocarditis, and pericarditis. And we want to study how genetics impact vaccines-related adverse reactions, which will aid in the design of newer and safer vaccine candidates. The study design that we propose for this project will be a case control study over a 3-year study period. For our study population, we will include healthy COVID-19 vaccinated people at any age, so pediatric and adulthood will be included. From vaccinated individuals, what we need is informed consent that if they don't give us this consent, cannot be included in this study. Cases or patients who receive COVID-19 vaccine and develop these adverse events of special interest over months after vaccination. We really need case validation for these cases. Controls are participants who received COVID-19 vaccination and did not experience any events over 4 months after vaccination. The measurements that we need is we want the DNA extraction for genotyping. So for this study, we want to collect saliva. First, the AdversOmics study include only genomics factor. We were not going to go for metabolomics or other omics layer, which are very important as well, but we need blood sample for other omics layer. And just for the sake of ethical approval and also budget that we have, we want to start first with only genomics, and we want to only collect saliva from cases and controls. Other measurement that we need for this study, indeed, is like other studies that we need clinical and non-clinical measurements from both cases and controls. So as I said, for DNA extraction, we need saliva, and among both controls and cases, at any time we can collect this saliva and send it to a lab in Vancouver, Canada. The one important part of this research in this study is about the number of cases and controls needed for genetic study, which always is a case. We need to calculate sample size and power to get at least 80% power for this study. As we calculated, we would need 825 adverse event cases. For each event, we need 275, and controls will be 5,500 controls. Of course, as I said, this project is a collaboration with GVDN. We're not gonna have all these cases and controls from our side, but we can contribute to this study as much as we can. The assumptions that we made for this sample size calculation was based on the math, which is minor allele frequency set at 0.05, 0.10, and 0.15 with 1 to 10 case to control ratio. And the significant p-value were set like all GWAS studies that we run to 5 times 10 minus 8, which is based on Bonferroni correction for multiple testing. So the last part of my presentation is to start with discussion with you guys to how to get you involved in these interesting studies and what are the plan ahead of this research. What we want for this to get started is to get approval from ethical committee for our research proposal, which will be done locally. The source of study population is a bit of like the matter of discussion actually. Can we really reach to these numbers that I just mentioned, and how we're gonna do this? What are the sources of cases and what would be the source of controls for this study? And the final things, which is very important, is budget for this study. In a discussion that I had with GVDN, they said that for the saliva kits, which is saliva collector, and supplies and shipping, all will be paid by GVDN, but they cannot pay for personnel. So still we need to think and we need to brainstorm today about the planning ahead and the challenges that we have. And I would be very happy to have your suggestions, your comments, anything that you would think that it's useful for our discussion today and for getting started very soon. Thank you very much for your attention, and happy to take any questions that you have.
Thank you, Fariba. Very nice. Are there already questions from the audience?
Thank you, Fariba. Can we unmute everybody or not?
Yes, I have unmuted all, so they should be now be able to. Of course, you need then to unmute yourself also on your computer.
Are there any questions from the audience?
Or willingness to participate, because there is this, request now from the GVDN, as Fariba stated, to collaborate on this. Do people see an interest to participate in that and to collect the cases to identify and collect cases? And finding controls is not so difficult because everybody is vaccinated and most of us have no adverse events, so that's easy. But it's about finding the controls and assessing the phenotypes. Yeah.
Hello, this is Javier.
Mm-hmm.
Hello, can you hear me?
Yes.
Yes.
Okay.
Sorry, I arrived late. I had another meeting, and we are initially interested, but I have to discuss how to proceed with my team that has been listening the webinar. So initially we're interested, but we will come to you later and discuss what we can do.
Okay, nice. Do you have any suggestion for us about cases? How can we reach cases?
I just arrived 5 minutes ago, so
okay.
So Javier, can you go back, like in Fisabio, you can go back to patients? Is that correct?
We will need to go through the ethics committee, of course, and then we will go through the different hospitals. Yes, it is not so easy as calling ourselves to the patients, but we can manage.
Okay. Yeah. Good. And what about Nicoletta? I saw. Are you still there? So what about Verona and for example, the cohort event monitoring, or do you see ways that this can be done? We can't hear you, Nicoletta.
Hi, this is Alexia. I'm not really sure Nicoletta is connected since she doesn't answer. We haven't discussed this with Gianluca recently, so we might need to go back on this topic.
And the colleagues from CDAP or Valdebron, Uretain? Anybody from CDAP who could do this? So what is the situation in IDIAP? Like, could you go back to patients with approvals, of course? Hi, Maria.
Hi, I have some doubts about some of the variables because I'm not sure if we can capture all of them, but I can ask some of the CDAP staff about the availability of the variables.
Okay. Anybody else? Sorry. In the Netherlands, we will try to participate. So
Yeah.
and it's just, you know, it's more like a clinical study. So it's a bit different than just using health data, but at the same time, it's quite easy to take saliva. So it's not very invasive organizational wise. So. Okay.
I can't see who's in the meeting. Maybe
Yeah,
Daniel knows. You can?
Yeah, I can. So anybody else from, I think we have had all the teams. You did anything from Valdebron or from DSIU, Liz? Yeah.
Hi, this is Liz at the DSIU. It certainly sounds interesting. Can I ask, the saliva kits, can they be used by the patient at home, or would you have to see the patient in a clinic?
It can be done, yes.
So they can do it themselves, yeah.
Yeah, they can do it themselves.
Okay, I think I think we're certainly interested. I'm not quite clear about the practicalities of being involved, but yeah, we're interested, I think.
Okay, then the practicalities we need to figure out, but this is more like, is there an interest and is there capability to do this? Like, which source of patients would you utilize, Liz? Like, would it be CPRD, or you would go through more your own cohort event monitoring or so?
I'm thinking more through our own sort of cohort monitoring, but yeah, this is very early days.
Yeah, no, and this is where we need to think together. I have the same feeling, like it's easier because you're closer to the patient. They, you know. Okay. If there are no other comments or questions, I think this is very nice. Thank you. So, Fariba, do you want to say a bit more? Maybe I was late, so maybe you have stated that, like how you would like to proceed with this working group, or.
Yeah, actually, it depends on how much people are interested in this research proposal and they want to contribute to this study or not. If they're interested, then we can make a plan to have sort of like meeting to have more discussion about how to get started. And for the research proposal approval from ethical committee, we can discuss the documents that are needed for this, and also for the source of study population, we can discuss it within the group meeting that we have. But for now, I'm not sure how many people are interested, and if they're interested, what are the timeline for this study. We have to figure it out at the beginning of this study, of course. So just let me know, people can let me know by email as well. If they are interested in this study, then I will provide them any documents that they need to learn more about the practicality, everything scientifically about this project. And if I don't know about the answer to their question, I will ask GVDN people, because they're also on board in this study, of course. So just let me know. For now, I don't have anything to discuss more than what I discussed, of course, today. But yeah, just let me know if they are interested in participating in this research study.
Thank you, Fariba. There is also Nicoletta who wants to say something, I think, but
yes, please.
We have problems with the microphone, I think. Or is it working?
No, thank you. It's okay. Alexia already answered before.
Okay, good. Thank you, Nicoletta.
Because yeah, I can answer it.
Good. We will, of course, also support this from the Secretariat of WAC4U, and we will create like a working group on AdversOmics studies. So please, if you're interested, to participate and to be invited in follow-up meetings, please let me know or the Secretariat know. We will compile a list and send out information to those mailing lists that we receive. Yes, so please sign up.
This is great. Thank you very much, Daniel, for your support. This is gonna be a simple study, as Miriam said. We just need saliva collected from cases and controls. The only challenge is cases. Controls are, we know them. In the Netherlands, we've been thinking of like controls from our hospital and cases maybe from specialists from the hospitals. It's going to be a simple study, but the results will be very interesting.
Well, the interest is, what it will do is that we build up experience on exploring this route. And as you stated, Fariba, also the AdversOmics is really like a field that is coming up, because at the end, we are all interested to know why these, rare events happen to certain people. So
yes.
So because we are dealing with very rare safety concerns like TTS, myocarditis, Guillain-Barré. So what makes these people different? And it's probably something genetic or an interaction with the environment. So that is where we're trying to unite efforts. So that's why VAC4U is participating with the Global Vaccine Data Network, which are doing this for CDC or starting this up, and we are their European counterpart a bit in trying to organize this. So so there is a bigger collective working on this across the world. So
yes,
I think that's the opportunity to learn now that this can be set up. So there is a little bit of money to do it that we can distribute. It's not a lot, but it gives us, I think, the experience of whether this is possible or not. And no site in itself will be able to do that, in alone.
Yeah. I have a comment from Judith, who wants to look in the department further and know more about then practical things. Maybe Judith, if you share a list of interested people, we can also get directly in contact with them and CC you.
Because I think actually, as cases, we could also utilize those that are reported to the Pharmacovigilance Center.
Yeah.
Because it's about genetics, so the selection bias doesn't. I think there is like strategies to deal with equilibrium. So
yeah, we can do it at any time if there are exact cases.
Oh, sorry, it's Judith.
Yes.
Okay. First of all, thanks for explaining this great idea. It's very innovative. It has already been stated. And Fariba, I would be interested in knowing, some more practicalities to check if certain people of my setting could be interested, because here I know people also with knowledge on pharmacovigilance databases, and they have access to, and on some genetic stuff related to pharmacology. So I would be pleased to know further.
Okay, great. Just let me know what kind of practicality information do you need.
Okay,
and then
I will write you then.
Yeah, we'll let you know. Okay, great. Thanks you.
Thanks and congrats.
Thanks.
So Nicoletta and Alexia, will you discuss with Gianluca? Because I think it would be great that we can also report back to the GVDN who would be interested as VAC4U.
Yes, of course. We'll talk to him.
Yeah, great.
I think we can also share this information with the Pharmacovigilance Center. So
yes,
we'll come back to you. Yeah.
Very good. Thank you.
Good.
Okay. Anything else?
Have any other?
Yeah, we can close.
Yeah. Okay, thank you very much, everyone, for joining today's webinar. And I hope we will get you on board in this study.
Thank you. Thank you,
Thank
Fariba.
you.
Thank you. Have a great day. Bye.
Bye bye.
Bye.
Thank you. Bye. Bye.
Bye.
Thank you. Bye bye.